Hi, and welcome back to Intro to Biology for Majors, or BIOL 1224. This is your Chapter 5 PowerPoint. This is from the Openstax book, Biology 2E, Chapter 5, which is called Structure and Function of the Plasma Membrane, and this is the beginning of Module 2. Let's begin with our objectives. So we have 3 objectives here. We want to be able to identify the structures of major components of the cell membrane. We want to be able to identify the 3 types of passive transport, and we want to be able to identify the characteristics of active transport. So let's start the chapter by talking about phospholipids, and I do have a YouTube link here if you want any more information after going over this slide. If you're curious about anything having to do with phospholipids, that YouTube video will be really helpful. So the phospholipid is this entire structure on the image here. And a phospholipid molecule has a hydrophilic head, which would be this red circle, this red portion, and it has 2 hydrophobic tails. So if you remember from previous chapters, hydrophilic means water-loving. So this head, the red portion, is going to be water-loving, it's attracted to water. And hydrophobic tails repel water. They are not attracted to water. And you can think of hydrophobic, I always think of the word phobia, something that you don't like. So hydrophobic tails do not like water. So phospholipids actually tend to be arranged in this way on this image. They have the hydrophobic tails facing one another, and then they have the hydrophilic heads facing out towards the liquid or the water. So some of the notes that I have on phospholipids are that phospholipids are the main fabric of the membrane. They are, the cell membranes are modeled with a fluid mosaic model. The molecular structure and primary component of the phospholipid bilayer provides the function of the membrane. So the fluidity of cells is due to the molecular structure of the phospholipid bilayer, the cytoskeleton and the extracellular matrix. So when we think of fluidity of cells, think of the transports that we're going to talk about in the next few slides, so things moving in and out. So the most integral proteins are transmembrane proteins spanning the cell membrane from interior to exterior. So once again, these are just notes, and we're going to hit more on this stuff in the next couple of slides. So I do want to talk about a couple of different components. So first, let's talk about phospholipids. So that's what we're talking about on this slide in particular is phospholipids. They, once again, the location, they're the main fabric of the cell membrane. So you've also heard of cholesterol, right? So cholesterol is actually attached between phospholipids and between the 2 phospholipid bilayers. So this layer is on the outside of the cell, and there are different things kind of found throughout the layer of phospholipids. And cholesterol happens to attach between the phospholipids, so in areas such as this, or in between the 2 phospholipid bilayers themselves, so in areas like this. So next, let's talk about integral proteins. So integral proteins are actually embedded within the layers, so they're embedded within the phospholipid bilayers, and they may or may not penetrate through both layers. So they may be able to move, but they're located within those 2 layers. Something else to talk about is peripheral proteins. So they are on the inner or outer surface of the phospholipid bilayer. They are not embedded within. So peripheral proteins are always going to be found either on the outside or the inside of the phospholipid bilayer. They're not within those tail areas. And lastly, I wanted to mention carbohydrates, which we've talked about before. They are generally attached to proteins on the outside membrane layer. So let's just, for image's sake, pretend that the bottom here is the inside of the cell. This is the outside. So the carbs would actually be on this side. They'd be attached to the outside of the membrane layer. So now we're going to start talking about some of those modes of transport. So we're going to start talking about passive transport. And I like to relate things like this to real life. So passive transport, I think of passive as someone maybe who's not confrontational or someone who tries to take the easy way in conversations. And passive would be the easiest mode of transport. So passive transport, the 3 different examples we're going to discuss are diffusion, facilitated transport, and osmosis. And all of these occur naturally from areas of very high concentration to areas of very low concentration. So this image is actually a good example of that. This image is of passive transport. You can read that on the bottom here. So in this example, they're actually using some facilitated diffusion. The gray area here is going to be your phospholipid bilayer that we talked about in the last slide. So these hydrophilic heads and the hydrophobic tails. So in this example, I want you to consider the blue-black background here as maybe a water solution, and the yellow as being a cell. So these little items here are being easily diffused from areas of very high concentration to within the cell where there's a very low concentration. And this in particular is considered a hypertonic solution. That's an HYPER tonic solution, hypertonic. We're going to talk about different types of solutions. So basically what those cells are sitting in determines how that passive transport will happen. So let's kind of talk about that. Those are some of the notes that I want to hit on. So hypotonic, which would be spelled HYPOTONIC, hypotonic solution. That kind of solution actually has a lower concentration of molecules, and the cell in that example would have a higher concentration. So it would be the opposite of this image. So the water or the solution, the hypotonic solution, would have a very few of these molecules, and then the cell inside would have a very high level of molecules. And in a condition like that, water will actually move from this solution into the cell to the point of bursting the cell open. So then you can have something called an isotonic solution that the cell sits in. An isotonic is spelled ISOTONIC, isotonic solution, and it's balanced. So there is some movement of water back and forth across that plasma, or that plasma membrane, but it occurs, but its balance is maintained. So it's a very equal or very similar amount of molecules on either side. So then let's talk about what this image portrays, which is, again, that hypertonic solution, HYPER tonic solution, which has a higher concentration of molecules on the outside of the cell, and the cell itself has a lower concentration of molecules. So in this condition, water will move out of the cell and into that solution, and the cell will shrink and shrivel. So the plasma membrane, this gray area we've been talking about, is selectively permeable. So if you think of that, it means that it's selective about letting things in and out. So because some substances can move freely across the membrane, other substances are transported by proteins. So when molecules move across the cell membrane from areas of high concentration, like this one, to areas of low concentration, they're moving down the concentration gradient, which makes sense, because if you have high to low, you're moving down that concentration gradient. That is called passive transport. It does not require any cellular energy, and that's very important to know. Passive transport is one type of diffusion, so it requires no energy, no ATP, like what we talked about in the last chapter. So molecules move from areas of high to low concentration naturally, and once again, it's down the concentration gradient. When molecules move across the cell membrane from areas of high to low concentration, it's passive transport. It does not require any ATP or energy. So now let's talk about those different types of passive transport. But I do want to mention that the prerogative of a cell, or what the cell really wants, is that isotonic state. So it wants to reach a state of balance. It doesn't want that hypertonic or that hypotonic solution. It wants to reach a state where it could comfortably sit in balance. So now let's talk more about diffusion. So diffusion is a type of passive transport. It's a passive process of transport. A single substance tends to move from an area of high to low concentration, just like we said, until it is equal across a space. So until it reaches that area of equilibrium. So let's go into my notes a little bit. Simple diffusion occurs freely across membranes and follows the concentration gradient, which, remember, is high to low. So this image will show that to you. So this image depicts diffusion. This orange area here in the middle is going to be that phospholipid bilayer we've been talking about, that cell membrane. So there's an area out here in the, what you, I guess, would call solution, which is an area of high concentration of molecules. There's a very low concentration within that cell. So simple diffusion will move across that concentration gradient, follows it down, right? So it goes from high to low concentration. Facilitated diffusion is also free and follows the gradient, but requires that a protein be present across the membrane. And then there's something called active transport, which requires energy. It requires ATP. Movement of large molecules in vesicles is bulk transport. So let's break that down a little bit. So we have simple diffusion, which is what this image depicts. It has very freely moving membrane across the membrane from high to low concentration. Then we have facilitated diffusion. It's also free, which means it costs no ATP, no energy. And it follows the gradient, but it requires a protein, which would be located here within the cell. And it would then assist those molecules to flow freely from high to low concentration. But again, it has no ATP requirement. You don't have to have energy. So then you have active transport, which is the opposite of passive transport. And an example of it does require energy and ATP. An example of that would be moving large molecules in vesicles, and that's considered bulk transport. So whenever we were talking about the endoplasmic reticulum and the Golgi apparatus, we said that sometimes vesicles will carry proteins. And I kind of talked about vesicles as being like the Uber of the cell, right? So they pick up a protein at the ER and they move it to the Golgi. And to do that, to make it across a concentration gradient if it had to, or just to make it out or inside of the cell, they would require ATP and energy because it's considered bulk transport. So another note here, diffusion is passive transport that moves small molecules like oxygen gas across the bilayer. So diffusion is not moving large molecules and bulk transport. Diffusion is moving small molecules. And simple diffusion moves molecules, like we said, from high concentration to low with no energy, while facilitated requires the presence of a protein that spans the membrane. So it will sit within that phospholipid bilayer and assist with the moving of those small molecules. And osmosis involves the diffusion of water. So we're going to talk about that a little bit later too. And simple diffusion allows non-polar molecules, hydrocarbons, things like carbon dioxide and oxygen, and other small uncharged molecules to pass between lipids. Facilitated diffusion is also known as facilitated transport. And if you think about it, it makes sense to have a protein because that protein acts as the facilitator. So it's sitting within that membrane, that phospholipid layer here. And it is assisting or facilitating those molecules moving from high to low concentration, but it still requires no energy. So this is a good image of that facilitated diffusion or facilitated transport that we were talking about. So requiring that protein that sits within the plasma membrane or the phospholipid bilayer here. So facilitated transport moves substances again down the concentration gradient, so from high concentration to low concentration. Channel proteins allow some molecules to cross the plasma membrane. So it is selective, but these proteins allow molecules in to the cell. So facilitated diffusion allows polar molecules like water, glucose, ions, some sugars, and some larger molecules even can pass through these protein channels. So we were talking about simple diffusion in the last slide, and that type of diffusion only works with very small non-polar molecules like CO2, like carbon dioxide and oxygen. But facilitated diffusion can actually work with polar molecules like water, glucose, and larger ions as well. So transmembrane proteins are sometimes specialized to be transport proteins. They allow facilitated diffusion of polar, charged, and large molecules like we were talking about. There's also something called an aquaporin, and it allows water to pass into the membrane. So the way I like to think of it is it's spelled AQUAPORIN. So it's almost like the words aqua and a pore, like what you have on your face. So basically it allows water to pass in and out of that membrane freely. Okay, so now let's talk about osmosis. And osmosis, the definition that I have here is while diffusion transports material across the membrane within cells, osmosis transports only water across the membrane, and the membrane limits the diffusion of the solutes or the molecules in the water. So this image kind of shows what I'm talking about here. It shows osmosis in, the best way possible. So this yellow line in the middle of the beaker here kind of represents the cell membrane. There's an area of low concentration on the left and an area of high chemical concentration on the right. And then on this side, we actually show equal concentration. So that's the goal of all cells. Even though the water level, it differentiates in this, which isn't a perfect example, it does still show you that through that cell membrane, there's an equal amount of what they call solutes or molecules on either side. And that's the goal of osmosis. It lets that water freely move across. So osmosis is the diffusion of water through a selectively permeable membrane. There is a video too, a helpful video within Blackboard that you can watch, and that video shows sugar molecules, which are too big to fit through the membrane for diffusion. The water can pass through and will seek balance, and this process is vital to the life of all of our cells. So osmosis is the diffusion of water, once again, through a selectively permeable membrane. So now let's talk about active transport. So we've been talking about modes of passive transport. And like I said, passive, the word itself kind of means easy, not wanting confrontation. So whenever we talk about active transport, we're talking about actively trying to push molecules or solutes from one side of the cell to the other. So active transport does require energy, and like we've talked about, energy is ATP within our cells. It's like the monetary money of our cells. We have to have energy in order for things to happen. So active transport requires energy, it requires a membrane protein, and it moves against the gradient from areas of low to high concentration. So that's the main difference in passive transport in cells and active transport. Passive transport always moves the easy way. So it always moves from an area of high solute or molecule concentration to an area of low. And active is the exact opposite. So it's actively pushing from a low concentration to a high concentration. So some of the notes that I have here. Once again, active transport is the opposite of diffusion or passive transport. It moves molecules up their concentration gradients, not down. So this image will actually show you that. So this is the inside of the cell here. We have active transport, so we have energy pushing through this membrane protein that we talked about, which is within that plasma membrane, outside of the cell where there's actually a higher concentration of molecules. So it would be much easier and more passive for those molecules to enter the cell. But in this active transport, we're using energy from our cell to push molecules out. So bulk transport, we mentioned before, it has to do sometimes with vesicles. So some molecules are actually too big to fit through these transmembrane proteins, this orange oval here. Some cells can engulf really big molecules in a process called endocytosis, and they dump large quantities of molecules at once in exocytosis. So basically you have something like a vesicle pick up a large molecule within the cell, and then through active transport giving away energy, that vesicle will push those molecules outside of the cell to go where they need to go. And most of the time they're going to be carrying a folded protein that's going to go off into your body and do its job. So active transport is very important as well. So lastly, I want to talk about phagocytosis. So phagocytosis is a special type of endocytosis. So in the last slide, we kind of talked about endocytosis. This allows for large particles to be engulfed. So this is how cells of the immune system, for example, will destroy an invasion cell. So if you've ever seen like a short video of a cell grabbing a virus and engulfing it and eating it, kind of trapping it, that's called phagocytosis. So once again, our immune system relies on this for white blood cells to destroy bacteria and other damaged cells in our body. So this image really kind of shows you what it's doing. These would be then the invasion cells. This would be one of our normal cells with our nucleus. So it slowly engulfs those invasion cells and holds them within the cell and then actually destroys them and recycles the parts within the cell as well. Okay, so we've made it to our key takeaways. So there's 3 of those here. The first being that the cell membranes are primarily composed of a phospholipid bilayer, embedded with a variety of different proteins. So second is passive transport moves molecules from high concentration to low. Simple diffusion, which is a type of passive transport, involves free movement across the bilayer. Facilitated diffusion requires a protein because that protein is facilitating the movement that spans the membrane. And osmosis involves just the diffusion of water. So those are the three main types of passive transport we talked about. So once again, simple diffusion involves free movement across the bilayer with no help and no energy. Facilitated diffusion needs a facilitator, so it needs that protein within the phospholipid bilayer to assist with moving those molecules back and forth. And osmosis involves the diffusion of only water. So those are the ways to really keep those separate in your mind. And then you have active transport, and the third key takeaway requires both energy and a protein, and it's capable of moving particles from low to high concentration. It's also capable of moving bigger particles. So remember, passive transport was more for small molecules, whether they be polar or nonpolar, and active transport is really good for large or bulk molecules. Also, please remember that biology also involves, or always involves, carbs, proteins, lipids, and nucleic acids, and a lot of times those things are being moved within the cell. So that's the end of this chapter. Once again, this is the beginning of Module 2, so you'll have a few more chapters after this before you're ready to take another test. But thank you for listening, and again, follow up. If you have any questions, feel free to email me or watch the videos within this slideshow or on Blackboard. Thank you.